Hello, Professor Patrick. I'm here with my oral assignment. So my gene of interest is not a single gene, but an entire NH operon from Pseudomonas Stutzeri. The function of this operon is that it encodes 9 enzymes for the degradation of naphthalene and substituted naphthalene to salicylic acid. I have a hypothetical solution where an oil spill occurs in a hypersaline environment like Caspian Sea and Dead Sea. So as a potential remediation strategy, my aim of the project is to clone NH operon from Pseudomonas Stutzeri in Halobacterium noriscense, the extremophilic archaebacteria, by using a modified Pibello back 11 vector. The reason why I have decided to go with this vector is that it is highly stable with the longer DNA fragments, and it has Laxat alpha fragment that allows the blue-white selection in E. coli. I have already inserted the promoter region from XYLDEGF operon from Pseudomonas species, OREC, which will act as origin of replication in Halobacterium. It already has chloroamphenical gene, which will act as a selection marker both in E. coli and Halobacterium. So next, how I have inserted my DNA fragment in to back vector and why this strategy. So first, I have amplified my operon using long range hot star PCR. That is reason why I have chosen long range PCR is that the normal PCR do not amplify high fragments of DNA like this operon having a size of 11.6 kilobase species. Uh, After I get my PCR amplified product, I attached linker to the blunt ends of the PCR amplified product. Then I carry out restriction digestion of back and linker ligated PCR product using BAM H1 and HIN3. And then I identify my products using 1% agarose gel electrophoresis. Uh, uh, for extracting my DNA and back vector from gel electrophoresis, I have used QIAEX2 gel extraction procedure. And after I got my purified product, I have ligated the extracted bacterial artificial chromosome and my PCR product using Thermo Fisher T4 ligase. Next, how I have transformed the E. coli. Well, there are two methods, that is electroporation and chemical method. I decided to go with electroporation because it is highly efficient. The first step was the preparation of the electrocompetent cell. As I prepared the electrocompetent cells, then next step was the transformation of the E. coli. So for transformation, I thawed the cells on ice and then added 2 milliliter of DNA, that is back DNA, to the 40 microliters of cells, and then continued with the electric shock for a few seconds and got my electroporated cells. Then I added 1 milliliter of the SOC media and transformed the and incubated the transformed cell culture in SOC media for 1 hour at 37 degrees Celsius in a shaker incubator. And then I spread about 50 microliter of uh, transformed cell from the SOC media on an agar plate with 25 microgram of chloramphenicol and IPDG and XGAR, and incubated the plate for 24 hours at 37 degrees Celsius. The next was the how I have screened the colonies that contain my gene of interest, and how I continued with the quality control and validation. So as I already mentioned that the bacterial artificial chromosome had the Laxat alpha fragment. So the bacteria with my bacterial artificial chromosome having an insert will disrupt the Laxat alpha fragment, and the colonies formed will be white in color. So I picked up 5 colonies and grown them in the media, and then isolated my back in the DNA using Kiagen MIDI kit and purified it. Then after purification, I restricted some portion of the bacterial artificial chromosome that I obtained after the purification. And then I carried on with the pulse field gel electrophoresis of digested and the undigested bag. The reason why I chose pulse field gel electrophoresis is that the same as the longer fragment of the DNA. After getting my result on the pulse field gel electrophoresis, I went on with the sequencing, that is Illumina sequencing, to identify my back with the proper insert. So the last step was the how I transfected my Halobacterium noriscens. So for this, I use the gene gun method. There were other methods like salt in solid method and electroporation, but again, the gene gun method offers you high efficiency. The Halobacterium grown in pains medium with the maintained salt concentration were subject to the gene gun method, that is biological ballistics methods. The target cells fixed after the transfection were grown in a liquid media supplied with benzoate. Benzoate allowed the induction of XYLD EGF promoter and naphthalene and chloroamphenicol. To check the working of the NH operon, addition of FeCl gives the white color, which indicates that the salicylic acid has been generated by the activity of NH operon. So that's all. Thank you.